You have to submit now. Okay, it's already times now. It's already 8:08, so you can submit now. Okay, we will start our class now. Oh, we can't display the content. What is happening? Okay, waiting for the slide to upload. I think you can already download the slide, right? Okay, now it's uploading, I think. Okay, so today we are going to do module 5. So I already shared the slide with you. There will be some slight changes to the slide because it's shared up much earlier. But anyway, you look at module 5 that we are going to look at is structural organization of the cell membrane. Okay, the learning outcome. So if you have a module, you can actually turn to page 5, the learning outcome. You can look at it. It's basically discuss the significance of cell membrane structure by giving physiological examples. Okay, and then list down the function of the cell membrane and justify the importance in maintaining the life. And then we will draw a model of a structural cell membrane, indicate the organization of individual phospholipids molecule, integral, and also peripheral proteins, glycoprotein, glycolipid, and cholesterol in the lipids bilayer, and describe the structural organization related to the fluid mosaic theory or model. Then we discuss the interaction between the cell and also its environment, and then we discuss the pharmacological significance of cell membrane. Is that okay? Yeah. Anything, Muhammad Anip, you want to say anything? No.
No, doctor.
Okay. Basically, you can hear what I'm saying, right?
Yes, clearly,
Yeah.
doctor.
The first important thing we look at, discuss the significance of cell membrane structure by giving physiological example. So we know that basically the cell is surrounded by the cell membrane, okay? And we know that basically the interior of the cell is very much different from its surrounding, which means the intracellular, the cytoplasm, and also the content inside the cell are very much different from its surrounding. That means the external environment, or in this case, the human cell will be the internal environment. Sorry, Afkar, yeah, anything? No. Okay, if those are unicellular paramecium, let's say like protozoa, amoeba, protozoa that is swimming in a freshwater pool, then basically they are in direct interaction with the external environment. So the content of the cell internally will be very much different from those in the external environment. But for a human cell or those multinational, cellular organism, our cell are basically bathed by the internal environment, so it's surrounded by the internal environment, and yet the intracellular content is very much different from those in the extracellular or the internal environment. Okay, and why is that important? Anyone can answer the questions? Why does the cell need a cell membrane?
To control the substances that enter the cell.
Okay. Why is it important that you want to control, or the cell want to control the substances that enter into the cell?
So that the cellular activities are normal.
Okay, so the cellular activities are normal. Okay, the most important thing is basically you do not want anything that is going to disrupt the content of the cell, because the cell basically are maintaining a very extraordinary complex solution and constitute, which basically include myriad of proteins, nucleic acid, nucleotides, and a lot of sugars that the cell synthesize and accumulate, and also ions. Okay, the cell basically accumulates certain type of ions and also proteins and chemical reaction. So it's actually accumulating this at a great cost. So it actually use energy, which means it utilize a lot of ATPs to maintain this intracellular structure, their content that is different from the internal environment. Okay, so these are the most important thing that the plasma membrane, basically, or the cell membrane, that become the outer skin layer, basically the surrounding cells, basically surrounding, and it become a partition. And this membranes is basically impermeable to large protein molecule. As some of your friends says, you want to make sure that not anything can actually enter into the cell, and also those charged particles or those ions that are charged. Okay, so you want to basically ensure that the content in the cytosol are different, and it's also basically very selective to allow certain molecule or ions to pass through. Okay, so these are the significance. So if we can look at the example, for example, even glucose that is trying to get into the cell, it needs a glucose transporter. Why? Because glucose are large or macromolecule that is difficult to actually permeate in, across the cell membrane. So you need a transporter to be able to transport the cell across. And we also know now that basically the intracellular content for potassium is much higher compared to sodium. What happens if sodium get into the cell in large quantity? Anyone? And we know that outside the ECF, the sodium content is higher compared to the intracellular. So when large amounts of, yeah. Anyone?
It becomes more saturated, so the water content in the cell will decrease.
Okay. So if, let's say, as Akash, if the sodium content get into the cell, what happens if sodium content in the cell increases inside the cell or in the cytosol of the cell? Akash or anyone?
Osmolarity increase.
The osmolarity inside the cell will increase because we know that sodium basically has carry an osmotic pressure, so it's able to attract more water into the cell. What will happen to the cell then?
Hemolysis.
Yes, the cell will basically swell. That means it becomes bigger and bigger as water more and more accumulated, and then it actually breaks the cell membrane. So this is why the cell do not want the sodium to actually come into the cell. In the way, the cell also do not want to lose potassium. So we are going to look at why is it that the potassium is also important not to get too much outside the cell later when we do membrane potential. Okay, so these are a few examples, and then if let's say sodium able to get into the cell within a very short seconds of time or millisecond of time, the cell will basically extrude the sodium out from the cell, so that it's still able to maintain a different in ionic concentration for sodium. So very low sodium content inside the cell and very high potassium content inside the cell. So okay. Any questions so far? Basically discussing the first learning outcome about the significance of cell membrane. No. Okay, so I just move on to the next one. So you can basically read about the overview, which I already mentioned just now, just basically a brief one. And we know that basically the function of the cell membrane, so you can refer to the slide number 3. So because it has a unique structure, so the function is basically to protect the internal organelles and also the internal membrane, basically also compartmentalize the intracellular or the organelles into different microdomain or subcellular domain. Okay. And the other function is regulate the passage of substances in and out of the cell, which we already discussed, basically to make sure that it allow only certain molecules to get into the cell and not any other molecules. Yeah. And number three, because it has certain receptors on the cell membrane, so it's able to detect ligands or chemical messengers that arrive at the cell surface, and these are very important for cellular communication. Okay, that we are going to do in module 7. And then it can fuse the membrane with other cells to form specialized junction. Okay, example, the gap junction, we know that they are very important in certain cells for cell-to-cell communication and also for the exchange of ions. Okay, then number 5, it provides a passage between cells for certain molecule, especially we have mentioned just now, there's the gap junction. And also number 6, it forms a stable site for bindings and catalysts on enzyme, and that is what we are looking at. Basically, they are very important to make sure that the enzyme content are within a very stable. Yes, I think. Okay, and then number 7, especially you look at the mitochondria, it has an inner membrane that basically provide. You look at the inner membranes, basically it has an anchoring site for cytosol, basically to make sure that the extracellular matrix, the components basically can actually link to that, and it has certain contact with the outside. Okay, and also finally it allows the cell mobility. That means it can actually move. When you look at the macrophages, okay, or the monocyte, when the monocyte get into the cell, it becomes macrophages and start to form pseudopores for it to be able to move inside in between the cells or in the tissue. Okay, so these are the significance of basically the cell membranes. I already give you an example. You have any question about that? Basically, answering the learning outcome number 1. Is there any question on this? Nobody put a hand, so I believe none. Okay, so I'll move on. Now we look at the microstructure of the cell membrane, and we know that structurally the cell membrane is not static, which means that it's just not like a piece of plastic or something that cover the cell. It's basically dynamic, and we know that basically it can be actually influenced by physical and chemical factors, and the movement basically also influence that basically provide an optimum physiological condition. And we know now, based on the fluid mosaic theory, the cell membrane has a protein molecule that's embedded in the lipid bilayers. Yeah. So we look at the cell membrane structures. Okay, this is one of the examples. I just want to draw. I will just stop sharing. You can refer to your slides that you have. Still okay? I will just on the. I have stopped sharing, right? Stop presenting. Yeah. Okay.
Dr. Hafiz, you are muted.
Sorry, I'm muted.
Okay,
Is that
now
okay
we can
now?
hear you. Yes.
Yeah,
Thank you,
yeah.
Doctor.
Thank you. Okay. We know a cell basically we tend to draw the cell around one, and that is basically easily understood. And we know that it has 2 layers. Okay, and in order to look at these microscopic structures, we know that basically it's composed of lipid bilayer. Okay, and also lipid bilayers. Okay, so the cell is basically have a cell membrane that is composed of lipid bilayer. Let's say you take a phospholipid, and we know that the phospholipid is composed of a head and also the tail. The tail is basically the fatty acid chain, and this head is basically the phosphate head. Yeah, let's see. Okay, and now if you take a beaker, okay, and this is basically water. What happens if you pour a solution of phospholipid onto the water? Right. The funny thing about this phospholipid is basically it do not light. I just write this off. It doesn't light. We expand it. It doesn't light the water. So what does it do? It basically convert its head, which is hydrophobic. Sorry, yeah, and also the tail, which is hydrophobic. So the tail. Different color. Okay. So on the surface, basically because it's a lipid, so it's basically lighter than water and it's going to float. And when it float, basically the head that is hydrophilic is going to basically attract to water. That means this is attracted to water molecule. So it's basically attracted to water. So the head will basically facing the water phase, and the tail will basically is facing outward into the air. Right. So this is the problems of phospholipid. So because it has the head that is hydrophilic and the tail that is hydrophobic. Okay. Now what happens if you take this beaker and you shake it? What will happen to the phospholipid then? Now, if you are able to mix and try to mix the phospholipid with the water, now it's basically that the phospholipids, rather than just if it is able to break into small particles, it will basically have, it started to form a micelle, and it has the head that actually pointed towards inside the micelle. Right. So basically, it has the tail that actually pointed inside into the micelle. Is that okay? Anyone? Anything you want to ask? Yeah. Somebody want to ask something? No? Okay. So we basically know that this is the problem with this is basically we are still in the water phase. The water phase is the one that basically this is attracted to water. So that's why it form a micelle. Okay. But if let's say this micelle getting bigger, what will happen if it start to get, let's say when you shake, some of the particle become bigger. If it's become bigger, then it start to form structures that the head will basically surround the micelle, which is still the same, but now it starts to form a bilayer in the sense that basically the lipid tails, the tails now facing each other to form a bilayer. So you can just basically draw the whole thing around. So this is how finally it becomes a bilayer. Yeah. Is that clear? For the phospholipid, why does it form a bilayer in this case as the micelle become bigger? So rather than just the small one, it basically has only a single layer. Now as it becomes bigger, it becomes a 2 layers. This is what happens in our cell membrane. Yeah. Because our cells is in a our cell is basically in surrounded by is water based. The ECS is basically extracellular fluid or the internal environment. Is water based, basically a lot of water. So the cell, there's no other way the cell membrane can do is basically form a 2 layers. So that there will be a content inside there, the cytoplasm and other molecules, organelle that can be actually stored inside the cytoplasm. So that's why it becomes a 2 layers. So you have the head that is facing the aqueous phase and also the tail that is facing inside to each other, okay, forming the lipid bilayer. Is that clear? Anyone? If you have any question, I think that is quite clear, so you don't have, so I just move on. Right. So what happens? Yeah, any question?
Doctor, can you explain this sphere again, this cell?
The cell.
Yeah.
Okay. I will just rub it away again. I say we start with a beaker of phosphor or water, right? So if you just put in phospholipids into the water, so what will happen? It basically is going to form a single layer with the head that is in contact with the water phase and the tail that's in contact with the air. Because the tail is hydrophilic, it doesn't like water. Sorry, it's hydrophobic, it doesn't like water. Yeah, the head likes water, so it basically interact with the water. Is that okay? But when you take this beaker and you shake it, what happens when you shake it? Because basically you're trying to mix the phospholipid with the water, and it's not going to be able to mix that content. Okay, so that's why you start to form micelle, which have the head that is facing still the aqueous in the water is still the aqueous phase, but the tail is projecting into the center of the micelle because it doesn't like water. Is that clear? Now, as you shake more stronger, or happens basically now this micelle sometimes they coalesce with other micelles, okay, other structure, and now they become a bigger micelle. So in order for a bigger micelle to be formed, there is no other way. Is basically you has the tail into the layers, and then you have the lipid layers. Okay, that's how it's able to form a slightly bigger structures. As we say, as it become bigger, the micelle, it can become 2 layers. Now here you have the aqueous phase, there's the water phase, and you have the external environment or the internal environment is basically the aqueous phase. And these are the heads that is in contact with water, and over here the same thing in contact in water. Okay, so until this stage, any questions? Yeah. Now, how does the cell able to then get bigger? Rather than just based on the micelle, it has to become bigger to be able to accommodate all the other organelles. that is how, as the cell become bigger, okay, now the bilayers, basically the head will be over here and the tail will be facing each side, just like the my cell getting bigger. Now you have cytoplasm, organelle, okay, and all the other organelles. So in order for the cell to get bigger, but the cell cannot get any bigger than it can be, as I discussed in the first module. Why does a cell cannot get any bigger or bigger than the egg or chicken's egg? It becomes microscopic. Why? Is the plasma membrane the limiting factors? The cell size. Why is the size of the cell is microscopic? Anyone?
The total surface area to volume ratio.
Yes, because the surface area that allow diffusion of ions or exchange of oxygen and carbon dioxide, basically that actually limit the size of the cell. It's not the plasma membrane or the cell membrane that limits the size of the cell. Yeah, so it's the surface area to volume of the cell that actually limits it, because most of the organelle cannot be too far away for diffusion of these important chemicals, diffusion for these chemicals to occur. These chemicals cannot diffuse too far away from the cell or from the organelles or organs that require this. Okay, so that's why it's limiting. But the cell size can become bigger than a single micelle, become a bilayer. So this is the structure of the cell membrane and why the cell membrane able to become bilayers. So we know now the cell membrane is basically a bilayer. You have a phospholipid that is facing each other. The tails is facing each other because they don't like water. Okay. You can continue drawing that. Okay. Question is, if let's say the cell membrane only have phospholipid as the only content, what will happen to the cell then? Imagine that the cell membrane is only phospholipid, 100% phospholipid. What will happen to the cell then?
Does that mean only one layer, doctor?
2 layers. Okay, I can just draw 2 layers. The cell membrane is 2 layers, but it's a phospholipid only. What will happen to the cell?
No organelles inside.
Yeah, okay, you can have organelles. Okay. Any other things? What will happen to the cell?
Like it won't receive certain molecules or ions.
Example of molecules or
'Cause it's-
ion.
Yeah.
Okay. Will you think the cell in this case is able to survive?
If it's
Will the
phospholipid only, that means that in between the phospholipid, there is no cholesterol to hold them together. Is it that way?
Okay. Yeah, just phospholipid basically part is also cholesterol. It has only 100% phospholipid. Well, if you want plus cholesterol, the other. So basically is a lipid base. What will happen to the cell? Sorry. Okay. Can oxygen get into the cell?
Yes.
Yes. Can carbon dioxide get out from the cell?
Yes.
Yes. Now can potassium get into the cell?
No.
No.
No,
There's no
because it's charged
protein.
ion. Okay.
Yeah.
So can sodium get into the cell
No.
or come out from the cell? Basically can't, right? Can glucose get into the cell?
No.
Yeah, basically no, because glucose need a transporter, so it can basically not get into the cell. So if the cell membrane is 100% phospholipids, only ions or chemicals or molecules that are soluble in lipids can get through, but water can usually also get through. Okay, because water is basically a neutral molecule, so it can easily also get through, and also on small size. But it cannot get in glucose, so the cell is not going to survive, right? Because it needs glucose as an energy source. It can also break down some fats and lipids, everything to generate the energy, but it will be very difficult. Yeah. So in this case now, the cell becomes more intelligent and says that I cannot just have only phospholipid as my cell membrane. It's not going to work for me. So I need protein to be in there. So it's embedded with proteins. Yeah. So that protein basically helps to create a channels that allow glucose to get in, let's say, okay, or even basically, okay, you have your sodium potassium pump that basically help to pump out sodium and take in potassium. So that's why now the cell has a protein that is embedded in it, in its phospholipid bilayer, and this is where we know then as a cell membrane with phospholipid protein embedded in lipid bilayer. Is that okay? But some cell actually has also carbohydrate, not just phospholipid, not just protein, but majority of it is protein. Some cell, especially our epithelial intestinal cell, epithelial cell, they basically has carbohydrate, which is on the surface. Or most of the cell actually have carbohydrate on the surface, but carbohydrate doesn't stay as carbohydrate itself. It basically binds with either lipids, become a glycolipids, or it binds with protein, become a glycoprotein. Yeah. So we will see later why are they important. Now, the lipids that is embedded in the cell they are on a static state. That means they are not moving, just like the tadpoles. Are they just stay static to one place?
No.
Yeah. What is the justification that they are not? Okay. Now we know that for basically the lipids bilayer, they can be a solid state, or they can go into a gel state. So what does that mean when we talk about the solid state or the gel state? Which means that this phospholipid can actually move within the plane of the cell membrane, right? Which means that the phospholipid by itself is not static. It can actually move. But in order to move, because all these molecules, they are basically embraced by the neighboring lipids molecules. So the phospholipid basically embrace each other, so they have difficulty to move from the single plane, the same plane, okay, this way. Yeah, it can actually move within the plane of the cell membrane. Yeah, but it cannot flick over the other side. That means from here, it flick over the other side and stay here. So most phospholipid is not able to do that. It basically sticks to one plane. If they are those facing outside the cell, so they will be basically moving outside the, on that plane. If they are facing inside the cell, they will only move facing inside the cell. Understood? Yes. No response, I assume that you understood. Which means that in order for this cell to be able to move within the plane, there must be increase in energy. And this is basically the thermal energy. So if the temperature of the surrounding the cell, if the temperature increases, okay, this phospholipid molecule will be able to gain more energy. As it gains more energy, it will basically breaks away from the friends that is holding it from there, and it start to move. So the cell membrane, as the temperature increases, it becomes more basically like liquids, basically more liquid. But as the temperature reduces, these molecules now become static because it's not gaining enough thermal energy to move with the plane of the phospholipid. So basically it becomes solid. Okay, so the cell membrane itself can become very wavy as the temperature increases, or become very static, become very stiff. Okay, because it cannot move as the temperature reduces. So the phospholipid is not able to move around. Is that okay? Why is that important? Now, if you relate that to the RBC, and we know the RBC or the macrophages, okay? Macrophages, they basically will move between the cell. So I just to rub this off. Okay, within the cell, the macrophages will basically move and is able to change each cell membrane, the shape of it. Okay, and when you compare to RBC, it basically have to squeeze through the capillaries, which is usually smaller than each cell. Okay, so it have to basically screw, okay, and basically distort its shape. If it's too solid, become a gel state, it will basically going to break the structure of the membrane and it's going to cause lysis. Is that understood? Or what happens if it becomes too liquid? If it becomes too liquid, it might not be able to hold the content and it can actually also spill out the content. So either way is not good. So either it becomes too so or too liquid or too solid to become too gel. So over the extreme here is also no good. Over extreme here is no good. So it's basically the best way is still in between. That's why our body temperature is maintained at 37 degrees C or 36.5 degrees C because of the structures of the cell membrane at between the so and the gel states. Is that understood? Any question? No? Okay, good. So everyone understood. Now, as we know, can you turn to page, not page, I'm using the slides. So you can look at the slide number 12, look at cholesterol then. But the rest of phospholipid, you can read on there, yeah? You have different type of phospholipid, basically. Okay, it depends on the content. So there are inositol, you can serine, you have choline, okay, on the head. So that determine the different type of phospholipid. And also you have the length. If you look at slide number 9, yeah, the length of the fatty acid, it can be very, very long chain. It can be as long as 14 to 18, even longer than that. So the longer the chain, okay, it makes the cell become less movable. Yeah. So the fatty acid change also can also determine the fluidity of the cell membrane. Okay. I think I have already done that. Okay. Now, in order for the cell membrane to be able to maintain its position or the static position, so if you look at cholesterol on the slide number 12, okay, the good thing about cholesterol is that basically it helps to basically anchor the cell membrane, the phospholipid layer, okay, so that to have cholesterol here, okay, that basically prevents the phospholipid molecule from moving within the plane. That basically make the membrane more rather than too fluid. Is that understood? Okay. We always bring cholesterol there. It's not a good thing because we say that cholesterol is bad for our health, but in actual fact, the cholesterol is very important basically to give the cell membrane a more solid state rather than too liquid. So if 100% liquid phospholipids, then it becomes a problem. So this basically help to fix the cell so that it become more static, more solid states in the way. Yeah. But the advantage about this cholesterol is it can flip between these two positions, which means that this cholesterol molecule, if they are here, it can actually flip over and move out. Okay, which means that it can actually go through the cell or it can actually go out from the cell. And these are very important because the cholesterol is the one that we use to generate steroid hormones. Okay, for our hormones production. Yeah. Because they basically is a precursor for steroid hormone production. Or if there is anything wrong to the cell, so basically if they don't have enough cholesterol, then you are not going to produce these steroid hormones, which is basically our sex hormones. Then it's going to affect our reproductive system. Is that okay?
Doctor?
So if you look, yes.
So doesn't it function to secrete steroid hormones outside of the cell membrane?
Yes, but you need the cholesterol for the cell to be able to produce that. You need to be able to take in the cholesterol first. So the cholesterol, basically, a lot of that are basically present on the cell membrane. And this is basically able to come into the cell to become the substrate for the cell and to use it to basically produce steroid hormone. Okay, the most important thing, you have all the sex hormones and also the cortisol and also for the aldosterone. And we know the importance of this hormone later on when you do endocrinology. Yeah, other than the sex hormone, basically testosterone and also the estrogen and the progesterone. So, okay?
Yeah, I understand that point, but I mean that if it flips, how does this affect the secretion of steroid hormones?
Steroid hormones is easier because it basically, they don't need any transporter. It basically just diffuse. When it's being formed, it will diffuse out from the cell, go into the extracellular fluid. And the problem is this, then it need to have a carrier protein to transport it because they are steroid. They are not soluble in the liquid or the aqueous phase, the water phase. But it's easier for this steroid hormone to basically diffuse to once they are formed inside the cell, they can just diffuse through the cell membrane into the cytosol or into the ECF for transport. Is that okay?
Yes, I understood. Thank you.
Okay. Yeah. Okay, so that's about cholesterol. Sphingolipids. Okay, I already discussed with that. Now we look at we'll come back to the modules. Okay, so we look at the modules. Look at page 9, activity 5.1. Okay, yes, Khairo. Sorry, is there any questions, Khairo? No. Okay, we just look at activity 5.1, cellular organelles and cellular chemical reactions. Okay, you can now list down the chemical reaction that occur inside the organelles and look at the function and how are plasma membranes enclose each organelle involved in regulating the cellular chemical reaction. So it's provide you the hint, basically transport of substrate and also the end product, and also there are present or receptors that can be activated by the hormones, okay, and neurotransmitters. So these are very important, basically the function of the cell membrane. Yeah. Then how are endogenous chemical or hormones, neurotransmitter hormones, factors can basically affect the cellular chemical reaction? As we know that basically the structure of the cell would depends on the protein, the type of proteins that is present on the surface of the cell. I have not covered the protein yet, so I do not want to go into that detail yet. That assuming that you have a cell that it can respond to steroid hormones. So example, let's say aldosterone. We have been talking about aldosterone, which you know is a steroid hormone. When it get to the cell, okay, it basically diffuse. This is not. It can easily diffuse through the cell, and it has a cytosolic receptors. Why is the aldosterone receptors is found inside the cytoplasm, not on the surface of the membrane? Why?
Because it cannot pass through the cell membrane because of the phospholipids, so it needs to pass through the cytoplasm, like starting from the cytoplasm.
Okay, it is good try, but that is not the right one. Basically, we know that aldosterone is a steroid hormone. When we talk about steroid hormone, that means they are lipid soluble. If they are lipid soluble, basically they can pass through the lipid bilayer without the need of protein carrier to transport it across. So if you have receptors that is present on the cell surface of the cell membrane, the aldosterone or the steroid hormone is not going to find it there because it basically diffuse through the plasma membrane or the cell membrane or phospholipid bilayer and look for the receptor in the cytosol, in the intracellular. Is that okay? Is that understood? Adil?
Yes, doctor.
Yeah. So that's why for steroid hormone, most of the receptors, they are found inside the cytosol because they can diffuse through. So they do not need a surface receptors or for it to basically respond to it. So this one, it can basically directly affect the gene synthesis in the cell. And then from there, is able to do all the necessary function. And one of them is basically to excrete potassium and transport sodium, okay, into the cell or basically across the cell. Yeah. So this is the function of a phosphoaldosterone. Okay. So I have covered that, and then when we look at proteins later on, then we look at cell signaling. Yeah. So you turn to page 10, plasma membrane. Okay. Why are the cellular chemical reaction important? You already know, right? Is there any question on activity 5.2 that you want to ask? Because some people has asked me how to get basically how to get answers for your the modules. Yeah. Any questions on activity 5.2? If not, I just move on. If you have, then we'll discuss.
Doctor,
Yes.
The second last question for 5.2.
If the thickness of the cell membrane is 10 nanometer, is that right?
Yes, that's the question.
Yeah. The question is, if you have 10 nanometer and the thickness of single paper is 100 micrometer. So one cell membrane is basically 10 nanometer, okay? And the thickness of the membrane is basically 100 micrometer. Okay, question is, how much is 1 micrometer will be equals to? 1 millimeter will be equals to micrometer. usually is 1000, right? Then 1 micrometer will be equals to 1000 nanometer, right? Which means that 1 millimeter will be equals to 1 million nanometer. Still okay? So now you have 100 micrometer will be equals to 100 times 1 million divided by 10. So you got probably about 10 million of the cell membrane can be stacked on the thin sheet of, let's assume that your size of paper there, then that's the amount that you can actually stack it out. Still okay?
Doctor, my question is, can the cell membranes be stacked on top of each other in the first place?
Yeah, in the first place, we put an assumption for calculation purpose.
Ah, I see. Okay.
Yes.
Thank you, doctor.
This is just for calculation purpose so that you can assume that how thin the cell membrane is based on the paper that we actually use every day. Still okay?
Yeah, doctor, understood. Thank you
Okay.
very much.
Okay, thank you. Any other questions? If not, I just move on to the next one. So if you look at activity 5.3, function of the cell membrane, now you can actually list down. I already given some of that, and you can actually find the answers in the module that the function of the cell membrane. Yeah. Is there any question you want to ask on this 5.3? No? You're okay with it? Okay. So if that's the case, then I will just move on to the next one. That will be microstructure structure of the cell membrane, which I already discussed in quite detail. When we look at the model, activity page 16, activity 5.4, model of the cell membrane. I already basically draw on the board just now, and also looking at the structure, three-dimensional structure of the model. And we have actually, it's not really three-dimensional, we put into two-dimensional. Any question on this activity 5.4?
Doctor, what are the improvements for the second question?
Improvement, okay. Make improvement on the model as you comprehend more information about membrane from this module and subsequently modules, yeah? Which means that you can actually add onto the model. Okay, initially we just have a plasma membrane. This is two-dimensional, okay, and you basically just add protein, okay, or you add, there's allow it to transport, and you only basically add only proteins, okay, or sodium potassium pump. Okay, so can you add more improvement as you go on? Now you have receptors. Let's say when we do intracellular communication, then you have receptors. Then you look at how the proteins that span, your G protein. So you can actually add on to that model. Okay, and how that actually affect non-cyclists, let's say. So this is how we can add more as you go through the module. Currently, you only have the idea, basically, it has a phospholipid and there's a protein. Is that okay, Adil?
Yes, thank you.
So as we progress on, we can actually add on to the model more and more information, especially looking at the voltage-gated channels, and then from there you look at why the membrane becomes excitable and non-excitable. Yeah. Okay, so I can just move on. Do you understand about the fluid mosaic theory now? Do you understand the fluid mosaic theory? Is there any question on that? You are okay with it, the fluid mosaic theory. Okay, so I just move on. If you understood. Then we go to the general molecular structure of the cell membrane, basically data interpretation. Okay, now if you found that from the analysis of the cell plasma membrane, and you found that it has more protein molecules, or it have more lipid content compared to the protein, what do you think the type of cell it would be? Okay, if you do an analysis of the cell membrane, and from this analysis, you basically found that there are more lipids compared to protein. So we have more lipids here, less protein here. Right. So what likely the cell will be? Anyone? Any idea?
A myelinated nerve cell.
Yeah, it's likely because this cell is going to prevent conduction of electricity, or there's not non-conductible. So it's likely to be the cell has a lot of myelin. If we count a myelin sheath, basically is wrapping around the axon. Okay, so it has this myelin. So if you analyze this cell, it will has more lipid content compared to protein content. So if you analyze this, then you will know that basically the main purpose function is basically to prevent conduction of electricity or action potential or membrane potential, basically. Yeah. So that's why, as Waka says, they are mainly for insulation. Yeah. Okay, then you can go further from there and look at why and how are some of the outer membrane. If you look at mitochondria, it basically has more protein content. So now if you analyze the membrane of a mitochondria, it's the other way around. You have more protein compared to the lipids. Yeah. So okay. So from there, you know that basically this have a lot to do with the enzymes or that is present on the surface of the mitochondria or the cell membrane of the mitochondria. The organelle membrane of the mitochondria. So it basically undergo a lot of chemical reactions that require a lot of enzymes. Okay. So what can you conclude from this activity? We already made the conclusion just now. So we went to activity 5.6, general concept of molecular structure of the cell membrane. Is there any question you want to ask? Okay, so this is more applicable in the sense that you look at the first question, how do the molecules, lipid, carbohydrate, protein become useful in carrying out the function of the cell membrane, which I already explained just now. So basically with the lipids, it prevents those large molecule and not lipid, non-fat soluble molecule from crossing across the membrane. Okay. And with the protein, it helps to basically transport this charged ion across. All right. Now, why are membranes said to be selective permeability? Anyone want to try that? Can you turn to page 19? Yes. Rania. Anything, Rania? I just read the message. Rania, have you anything you want to ask? No, I didn't hear you. Or can we take 5 minutes or 10 minutes break now? Because I think most of you go and wash your face, go and drink some coffee, and then we can come back again.
Yes, doctor, okay.
Yeah, just go and wash your face and drink some coffee because it was a bit quiet. So I don't know what happens. It seems to be a bit tired. We will start back at about 9, let's say 9:20. Is that okay?
Yes. Thank you.
So now I have only one student. I'll see you then.
Yes, doctor.
Okay. Okay, Assalamualaikum warahmatullahi wabarakatuh. And we start again. Is everyone in now? Can you put your hand that you are around? Yes. Good. Yeah, there's a lot. Many of you are around. So I thought I was only talking with Adil and also Waka. So I have only 2 students. Okay. So most of you are in. Helmut, can you send the attendance out for them to sign in, please? Oh, it's only half of you are there. Helmut? Yes, Akash, you have sent, you have done that? Yes, thank you. Thank you. Thiru, you were asking about the flipping parts. You want to know about how cholesterol able to flip between the 2 plane. That's what you're asking, is it? I will try to explain that again if you need to. If you understand that, then it's okay. If not, you have to explain that. Is Thiru in? Yes, Ani.
Yes. So, yes, doctor.
Do you have any question? Yeah, Thiru, you still want me to explain about the cholesterol that is flipping from the one plane to the other plane?
Yes, if you can.
Okay. I will explain again about the cholesterol, and we know the structure of the cholesterol. Okay, it's already 21, so we can start now. Okay. So if you look at the cell membrane, basically you are going to have this. As I told you just now, okay, so you will have a cholesterol molecules which are basically going to be this way, okay. As I told you just now, basically the lipids will basically only able to move within the plane, depend on the thermal energy that it has. Okay.
Excuse me, doctor.
Yes.
Doctor, have you started your whiteboard?
Yes, I'm sorry. I
Okay.
forget to on the video. Okay. Yeah. Can you see it now?
Yeah, we can.
Yes.
Okay. You see? Okay. Yeah.
Okay.
So if we look at the plane, you have phospholipids bilayer, basically is arranged in this way. So it depends on the thermal energy. The thermal energy. So if you have higher temperature, so this lipids molecule, phospholipid molecule is able to move within that plane, which means that if they are in, this is the external environment or the internal environment, okay, this is intracellular or the ICF. ICF, this is the ECF. So these molecules of plus phospholipid basically is going to only able to move this plane. Why? Because of the tail part of this are basically much difficult for them to flip to the other side compared to the cholesterol, because the cholesterol basically they are quite rigid. Cholesterol are rigid in the sense that you have the cholesterol molecule, which are basically the rings, right? Okay, and these rings are basically allowing okay, this is a cholesterol ring, which are basically quite rigid compared to a phospholipid. As we know, phospholipid basically you have the head and you have the tail. Okay, The tail, it depends on the molecule in the tail. So we tend to just basically draw them a straight line, or they may have a double bond, then they might have a king. Okay, but as a whole, the phospholipid tail are basically very long and flexible. So because of that, that's why they tend to be more liquid stage than actually in the gel state. Yeah, and they can only move this way because most of these tail are being, because if there's another one that the tails basically interact with the two of them, and they find it difficult to basically flip between them compared to the cholesterol. Then you have basically the tail. Okay, the cholesterol are basically, because of these four rings, it become very rigid, but because it's able to move because it only have a head of hydroxyl, which doesn't require very much energy, thermal energy, to basically flip. So it can actually flip between the two planes. Okay, that makes the cholesterol able to basically flip. Even though it's rigid, it can anchor all the phospholipid between them. Basically, it can make them become very rigid, but itself, it can fit to this different plane. Jiru, is that okay?
Yes, doctor. Doctor, if flipping, you mean by like they can make a U-turn, right?
They just change the position within this. That means those in the ECF plane, you can move to the ICF, or it can, either way, it can flip. For a phospholipid, it cannot. It can only move one plane. In the ECF plane, ECF plane, ICF plane, ICF plane, it cannot flip because of its tail, the fatty acid tail, which is long fatty acid tail.
Okay, doctor.
Okay. So we are finished with this. Now we turn to page 21. Look at activity 5.7. We talk about the membrane lipids. Everyone is still have you get your cup of coffee?
Yes, doctor.
Jasmine and also Kaswina, you have any questions? You can put down your hand if you don't have. Yes, Jasmine.
No question.
Okay, you are clear, okay?
Yeah.
Can you just put down the hand? Yes, thank you. Okay. Now we look at activity 5.7. Okay, you look at the question, how do membrane lipids contribute to the fluid state of the membrane? Anyone want to respond?
Can you repeat the question, doctor?
Question is in activity 5.7. The second question says, how do membrane lipids contribute to the fluid state of the membrane?
It anchors the phospholipid so that it doesn't expand when it's moving far away, and it doesn't contract when it's cold. It's like it maintains rigidity, something like that.
Okay, that is, you are referring to cholesterol, right?
Yes.
But this is about the membrane lipids. So how does the membrane lipids contribute to the fluidity of the membrane? Do you understand the questions? Okay. So it depends on the thermal energy of the surroundings. Okay, if the thermal energy, which means if there's an increase in temperature, it will become solid state. Okay, or if there is a reduce in temperature, it becomes a gel state. So it can move between these two, depends on the temperature of the surrounding that provides the fluidity. Okay, but the most important thing of that fluidity is that if there is a break on the membrane, what will happen then? If it's not able to seal back the breaks, okay, but it doesn't use like fibrin or anything that basically fill it. It's basically just the phospholipid rearrange themselves and actually seal the blockage or seal any openings that is present on the cell membrane. Is that okay? So that is very significant, which means that you have seen scientists when they do experiments on ovals are very big structures. I mean, it's basically a bigger cell compared to the normal cell that we have. And you have all the genetic material that's in there, okay? The scientists can actually poke a pipette to extract the embryo. It can actually draw the embryo out. And then when you take it out, you can actually move to another cell and then implant the embryo in a new cell. This is what the experiments done. Do you remember that how they create Polly, the sheep, Dolly the sheep?
Yes, doctor.
Yes, these are actually cell membrane. If you poke a pipette and you actually break this, okay, when you take out the pipette, the membrane just seal back the breakage. So there is no problem on it cannot heal itself. It basically automatically just the phospholipid rearrange back again to seal out the breakage. Okay, that's how it's able to transfer the embryo to another cell. This is the they say the eggs you can transfer to our normal cell or even skin cell, and from there it can develop into an embryo or into a sheep, into a dolly. Yeah. So a lot of experiments that has been done, but this is now we say that it's not ethical, so they probably stop doing. Okay, is that understood? So what is the significance of that fluid state?
Doctor?
Yes.
For the lipids part, temperature, you mean the external environment temperature or the body temperature?
When you talk about if they are unicellular, then they depend on the external environment temperature. But if you are multicellular, then you depend on the internal environment temperature. Okay.
Okay.
Yeah, okay. Yeah.
Yes.
So because of that, that's why our body temperature had to be maintained around 37 degrees, so that our cell membrane is not too liquid and is neither too gel. So it just nice at around that stage, so it doesn't basically become too liquid. Okay, what happens? The temperature increases, which means that this is going to shift and move around. Now imagine there is proteins there. Okay, what will happen to the protein? It's also going to move, but it can actually get inside the cell, or it actually go up because it's able to hold the protein together. So this protein basically is going to drop into the cell and become non-functional. And we know that the importance of this protein later on, that without this transporter, it's not going to do this job as a transporter on the cell membrane. Is that okay?
Okay, doctor.
Then we look at the second question. It asks, the membrane lipids are mostly bipolar. They have the hydrophilic and hydrophobic parts. What is the significance of this characteristic? We talk a lot about this. Hydrophilic, and this one is hydrophobic. This is really okay. So what is the significance of that? We said the phospholipid is an amphipathic molecule. Is it important? Do you have any idea? Anything tried? Yeah.
Is it because it allows the lipid soluble particles to pass through?
Yeah, we allow the lipid particle to pass through because it's hydrophobic. Okay, how about the hydrophilic parts? Okay, The most important thing of this arrangement is that you now can actually form 2 layers rather than just a single layer. Okay, what happens if the cell membrane is only a single layer? Okay, 1 layer, which means that the content inside there must all be lipid soluble. Is that useful in the intracellularly? This is ECF and this is ICF. If they are just single layer. They won't be useful because you cannot have all the chemical reaction going on. There is only lipids because the enzyme itself is liquid. I mean, it's liquid phase. They are water soluble, so they cannot function. Yeah, that's why the cell is maintained as a bilayer. So that here is an aqua phase, and here is also an aqua phase. That's the liquid phase. So now you have content that is enzymes that can be liquid soluble. Okay, I hope you get the ideas now. Then determine the percentage of composition. I think that's okay. Any questions so far for activity 5.7? No? Okay, so I'll just move on. Then we look at activity 5.8. We talk about phospholipids, and there are different types of phospholipid, yeah? And you can actually work on that. If you have any questions, if not, then I will just move on, yeah? Then we talk about lipids and also cholesterol. I already mentioned about the cholesterol. Then you can work on activity 5.10. Okay. Any question about the cholesterol? Okay. Can you turn to page 28 now and then look at cholesterol, especially on question number 3, the third one, the third paragraph. Okay. Cholesterol is an integral part of the cell membrane, yet we are advised to reduce our cholesterol intake for fear of cardiovascular disease. So you can explain that related to the formation of arteriosclerosis. Why are we advised not to take so much cholesterol, and how does that relate to arteriosclerosis? Someone put out your hand and answer it. Are you all still with me? Can
Yes,
I break
Doctor.
off? Okay. Can you answer the questions? Yeah, Adi or Ani?
The excess cholesterol will be deposited on the walls of the arteries or blood vessels.
Good. How was that going to be a problem?
It will limit the flow of blood.
Okay, it will basically going to limit the flow of blood. Okay, now we look at cholesterol. So there are 2 types of cholesterol that we talk about. They are the HDL and LDL. And mostly the problem is because of the LDL portion, yeah, that we are worried about. Okay, imagine that this is the blood vessels, okay, and you start to have cholesterol molecule that's basically deposited inside there. And what happens is basically your macrophages that's going to come and basically engulf this molecule, right? What happens to these macrophages? As it engulf, it becomes what we call a foam cell. We contain a lot of this cholesterol molecule inside there, okay? And this is going to basically stress the cell, and it's going to cause a lot of cytokine that's being released, okay? And basically it's going to cause a formation of a thrombus, okay, with all this cell content there. And it is going to basically prevent the flow of blood through the blood vessels. Right. So it becomes very narrow to basically flow through. And this, if this thrombus break, is going to flow down. As it breaks, let's say the thrombus is here. If it break off, so it becomes able to basically block the blood vessel and cause problems in the downstream. Is that okay? That's what Anif you are referring to, right?
Yes. Yes, doctor.
Okay. Thank you. So the rest you can do about familial hypercholesterolemia, which is basically genetically linked, and it can be a problem because they have too much cholesterol. So you need to basically treat the patient with the anti-cholesterol drugs or anti-lipids drugs. And now currently, commonly people are using statin to basically reduce the cholesterol level in the blood. Okay. And most of our cholesterol, where is it from? Is it from our food that we took in, or is it formed by other organ system? New one?
Food, sir.
Yes, Ahwan. Yes, it's from our food. Yes, Adil.
No, I was answering food. That's it.
Also, okay. So basically, most of our cholesterol we thought was actually coming from our food. Yeah, but as we age, it's our liver that actually generate a lot of cholesterol. Our liver actually produce almost 70% of our body cholesterol. Either it's required or not required. So basically, that's how statin works by actually inhibiting the liver from forming more cholesterol. So people who take anti-lipids, basically they are taking statins to reduce the cholesterol that is formed, and it can actually helps a lot to reduce the cholesterol level. Okay, is that okay? So I just move on to the next one. That is looking at the fluidity. Look at activity 5.11. You refer to local anesthetic and also the membrane fluidity. Okay, look at the first questions. Is there anything you want to ask now? Turn to page 30. Okay.
Yes, doctor.
So we know that basically, yes, please explain. Okay. We know that basically there is a lot of proteins that is there. Okay. These proteins, right, it form a channels. Okay. So the most important one we have now currently is the protein channels called potassium channels. Okay. And these potassium channels or leakage channel, right, allow basically potassium to, let's say here is ECF, yeah? And here will be the ICF. Okay, that allow potassium to actually move out to create a membrane potential. Inside there is more negative compared to the outside, which is more positive. And these are basically the phospholipids in between. Right. Now, when you give a local anesthetic, that is lidocaine, which is then exert the effect by basically increase the fluidity of the fluid, which means that you allow this, now this molecule, when you give lidocaine, this molecule to basically move. Okay, if it don't move, basically it's help to basically fix the protein. The phospholipid help to fix the protein. Okay, now when this one become very movable, the phospholipid start to move. What will happen to this protein molecule? What will happen to the protein molecule?
It will be unstable.
It will become unstable. So it cannot, because nothing's to hold it. So it will basically drop into the cell. If it drop into the cell now, you don't have these proteins there anymore. Right, as it drop into the cell. So basically this molecule, these protein channels or pores basically drop into the cell. And what you have now is you close. So there will be no potassium that can actually go out. Right, you will cause hyperpolarization of the cell. Okay, let's say normally the membrane potential is about minus 70 millivolt. Now you hyperpolarize it, it becomes, let's say, 90 millivolts. Negative 90 millivolts. So now the cell becomes difficult to depolarize. Okay, that's where when you give lidocaine or anything onto this, basically it prevents the neuron from able to form any action potential or able to transmit the information. So that's how it actually reduced or elevated the sensory nerve, okay, and basically block the sensation of pain by the neuron, carried by the neuron. Is that understood, Adil? Or anyone? Is that clear?
Yes, clear, doctor.
Yes. Doctor, I have a question.
Yeah, okay, yes.
You mean the protein will fall down into the ICF, into the cell, right?
Yes.
Is there any chance it will go back into the phospholipid bilayer instead of getting destroyed or unused?
It doesn't actually get destroyed. It just only sink into the cell, so it's not present on the cell membrane. Because when you give lidocaine or procaine, it made the fluidity, it made this phospholipid become very soul state, which means that it increased the soul state or fluidity, increase the fluidity of the cell membrane. So the molecules of the phospholipid start to move around very easily. So it doesn't hold the protein onto its plane. So this protein basically in a turn cross, it drops into something like drop ship, basically it drops inside the cell. So it's not present on the cell membrane, and this doesn't allow potassium to leak out. Okay, and this protein are usually the leakage channel for potassium. So it prevents that. Now the cell start to hyperpolarize. So okay.
Yeah, I understand. Thank you.
Yeah, thank you. Okay, so that makes the thing become okay. So you understood this, yeah? So I just move on. So that's how this local anesthetic is able to work by basically preventing the cell to become polarized or hyperpolarize the cell, so it make it the pain sensation in the sensory nerve cannot be transmitted. Yeah, so that's why the fluidity of the plasma membrane is very important to ensure that these proteins stays on the plane of the plasma membrane. It doesn't disappear. If it disappears, then the receptors or the transporters are not present on the membrane. As I asked you question just now, if the membranes doesn't have plasma protein, then it's not going to function. It's not able to do its function as a for transporters or even for intracellular communication. Okay, so I'm done there. Yes.
Doctor.
Yes.
Actually, where's the protein come from?
Where's your protein coming from?
Yeah.
Okay, a lot of these transporter protein are actually produced by the cell. Okay, which means that if you look at the cell, okay, it will produce its own protein and inserted on the cell membrane. Okay, so it inserted on the cell membrane, and this protein is actually formed from your endoplasmic reticulum Golgi apparatus pad, and then finally it go and then it translocate and fits onto the cell membrane. So if you look on the surface of the cell from out, you basically see a lot of protein that is basically stuck on the plasma membrane. Okay, there are different types of proteins there. So this is what we call the fluid mosaic. So it looks like a mosaic. If you have the top view, if you look the cell from the top, this is what you are going to see. Okay, how the cell basically look like with a lot of proteins that is stuck in there. Yeah, and these proteins is basically not static. Later when we talk about the plasma protein, they are basically also moving as they go about. They are not fixed to one place. Okay, so if you, I just want to show you about the plasma proteins, and if you can look at the slides that I sent to you. on diffusion of the membrane protein. Okay, there's experiments that was done by two scientists. You look at slide number 19. Have you turned to that? Or I will just share the slide. Okay, I will share you the slide. Okay, so we turn to slide number 19. It was a bit slow. Okay, you look at these slides. Okay, basically it says, okay, can you see my pointer? No. Initially, they use a lymphocyte. They started with a colored dye on the surface of the membrane, and then they force the cell to fuse together, and they look at the color of the after half an hour. They found that basically the color of this start dye, basically representing the proteins, are now dispersed evenly across the cell membrane, which means that these proteins, can we come back now? Which means that this protein doesn't fix to only one plane. That means if you found that they are over here, they can actually later on half an hour, they actually move to this side, okay? Or they can actually move to this side. So they are basically movable. They are not fixed.
Okay, doctor, understood. Thank you very much.
Okay. All these protein on the surface of the membrane are produced by the cell. So each cell have its own unique characteristic. They will produce its required cell proteins on the cell membrane. So if they require certain proteins that is needed, and they give the characteristic of the cell because it's able to produce that type of protein that is required on the cell membrane. Is that okay? So that's why if you look at the proteins, then they are not only specific to particular cell, but they are very specialized for that cell, because let's say you look at receptors for hormones, only certain cells have that particular hormones receptor, then it can only respond to the hormones that is being secreted. If not, it's not able to respond to it. We will look at later on intracellular communication after the transport of proteins. Yeah. So can we move on to the next activities? That is basically membrane carbohydrates. Okay. So we know that membrane carbohydrate doesn't exist just like a protein. Compared to the protein, basically it can just have only protein molecules, not any other things. But most of it actually attached to a proteins, or actually attached to a lipids. But they doesn't constitute very much. Basically, around 2 to 10% of that are basically the carbohydrates on the membrane. Okay. And majority of this, basically, if you look at RBC, 52% actually attach, only 8% are basically carbohydrate. Okay, 40% is lipid, but 52% are proteins. Yeah. Okay. Now, all carbohydrate of the plasma membrane, they generally face outward. That means they are facing the extracellular field. That is understood because they are mainly for you look at that, basically they are mainly for cell interaction, and also they function mainly for intracellular interaction and also interaction between cell and the environment. They actually interact with the neighboring cell, and also for receiving interaction with other cells. Okay, then you have the glycoproteins. Glycoproteins, they are basically the surface and they are very essential for virus cellular activity, including cell-to-cell communication and also cell matrix interaction. Basically, the main things for glycoprotein are mainly for cell recognition, okay, and cell adhesion. And because of that glycoprotein that attached to the protein molecule, this carbohydrate molecule basically gives the cell an identity so that your body immune system can recognize it and basically acts on it. If it's found that it's a foreign cell, then it will act on it. And it know that this is a cell, then it will basically spare it. Yeah. So mainly for identification. Okay. And also a lot of study has found that basically these glycoproteins are closely associated to human diseases like cancer, rheumatoid arthritis, and also some immune deficiency diseases. Yeah. Okay. Now, when you yesterday while you was talking about the people who are hyperglycemic or diabetic patient, they will start to have advanced glycated end products or glycation end products or H. These are the proteins molecule. So which means that on the surface this attached to a protein, you have the carbohydrate. Okay, and these carbohydrate basically are present on the surface, mainly for cell recognition. They are contain many of that galactose and all this. Yeah. Okay, but when you have an increased glucose level, okay, what happens is basically it's going to interact with the proteins here on the cell surface, and this interaction is going to form what we call the advanced glycation end products. Okay, this product is not a good product, so it's going to cause a lot of problems. Basically, it leads to the problems of micro and macrovascular, or it can also affect the nerve function. Okay, so this is also can also contribute to development of arteriosclerosis. It can actually damage the basement membrane of the glomerulus and basically damage the kidney and also result in kidney failure. Okay, so that is all about the glycoprotein, and then we have glycolipids. Glycolipids, then we are familiar with the things that glycolipids are basically the glucose or the carbohydrate portion attached to the lipids. This basically gives you the type, and we are very familiar with this, the blood type A, B, C, or A, B, AB, or O. And you already know that, right? So I leave that glycolipids. So you can work on the activity 5.13, looking at the type of blood that the person has. Okay. Is there any questions on that? You can fill out the table. So I just leave that. And then finally, you look at activity 5.14, membrane carbohydrates, and we already know what actually forms that. So I just leave that to you to read. Yeah. Then we are going to the membrane protein. Any questions so far? Everyone fall asleep?
No, doctor. No, doctor.
Okay, yeah, still with me.
No.
Okay. I think I want to take a break now for half an hour. That means we are following the timetable at 10 o'clock now. We will break for half an hour, and then when we come back, we'll talk about a lot about the membrane protein, and this is a big or huge topics that we want to focus on. And later on to next Monday, we will be talking about the transporters. Is that okay?
Yes, doctor.
Okay,
Yeah.
then
Yes,
you'll come back at 10:30, yeah?
Okay.
Okay, thank you.
Thank you, Doctor.
Thank you, doctor.
Okay, thank you. I'll see you at 10.